noticed that sometimes when you sneeze, you have a lot of snot and sometimes you don't? Or did you know that the average adult lung capacity is about 6 litres, which means our snot-producing capabilities are rather low in comparison to another mammal, such as a whale, which can have a lung capacity of over 1,000 litres. In fact, we can use information such as whale snot to provide a wealth of information such as DNA, hormones, and bacteria. But how exactly do you collect whale snot? Well, previous methods involved the collection of health from animals that had stranded, in which case their health was seriously compromised. And other methods involves the use of deliberately killing whales. And current methods involves extremely close boat approaches, where scientists use poles with a collection device and hold this over a whale and wait for them to breathe. But this can be dangerous, as an 80-ton whale could easily flip a boat. So to make collecting health information from whales easier, I collaborated with drone experts in industry, and together we designed and built a waterproof drone with Petri dish for collecting whale snot from northward migrating humpback whales off Sydney for an assessment of whale health. Once a whale, or pod of whales, was sighted, we launched a drone from the back of our boat. Then once in position, the Petri dish was opened. Then as a whale took a breath, the drone was flown through the densest part of the whale's snot. Immediately after, the lid was shut, securing the sample and minimising contamination as the drone was flown back to the research vessel. Overall, we collected 59 samples of whale snot. I then used forensic techniques to try and identify the types of bacteria living in whale lungs. And interestingly, I found these whales shared similar bacteria with Northern Hemisphere whales and some dolphins. But I also found that these whales are acting as massive mobile monitors of our ocean health as they travel from the cool feeding waters of Antarctica, past the beaches that you and I swim in, and all the way to the warm waters of Northern Australia. And then all the way back again each and every single year as they migrate. My research is revolutionising the way in which we collect health information from whales around the world. The use of drones is giving rise to a new era in whale conservation. And I'm not joking.
I'm proud of you, Vanessa. That was spectacular. Wow.
Just to continue with the puns, have you had any really sticky situations in your research?
Excellent.
Well, that's a really good question because we rate the amount of whale snot that we collect from a juiciness scaling, which is an easy way to refer to. For the scientists out there, that's 100 or to 150 microliters, but for the non-scientists, juicy being 3 and a juicy-less sample being 1, and in the middle, number 2 of juiciness.
I find it very hard to work out how you get snot without a nose, but putting that aside for a minute, I would have thought with so much water sloshing around, clearing out the orifices, that there would be very little difference between them. Is there?
Well, between each whale, is that?
Yeah. Do you get a range?
So the idea between this is the method behind my research was to try and understand, can we collect whale snot? Because there's a lot of other problems in terms of contamination. So we wanted to know that what we were collecting was whale snot, not just air, not just water. So we collected air samples as well as water samples in addition to that of whale snot. And we were able to show that it was quite distinct. But more specific in relation to your question, we can use the individual profiles that we can collect from whales of the population I'm working on, the East Australian coast, to then compare that to other populations around the world. So we would want to know maybe how's the health of our humpback whales faring in Australia compared to maybe in the Northern Hemisphere. But another way we can also increase the level of understanding is that we can look at whale snot in the migration phase, also in the feeding phase in Antarctica where the environment is likely to be much different, and then that versus the warm waters where they're breeding and doing a lot of fun things, presumably.
I know I can't resist asking a question on this. It's just such fantastic stuff. And that is, could you get enough of a profile to identify individual whales as well, so you know if they're going up and down, that you can catch the same one and see how their health profile changes with time?
That is such a good question, and this is a question I am commonly asked. However, the fortunate news here is that for the East Australian humpback whale population, their numbers are on the rise. So there's around 30,000 of these individuals. But that's the reason we're using humpback whales as a study species to learn more about other whales, such as the southern right whale here in Australia, which aren't faring as well. So if we were to collect the same whale snot from the same individual, that would be great. And we could do that probably by using very famous whales such as Migaloo, which is the all-white whale that many of us may have heard of. But in terms of going, oh, I've sampled your snot before, oh, I saw you last year and you had a lot of boogers to that time, I can't just do that, unfortunately. Thank you.
Thank you, Vanessa. All right, our next speaker, James Wong, has come a long way indeed from just across the quad. University of Western Australia researcher. He will talk us through breathing while you hop. How do kangaroos do it? Please make him feel very welcome.
So tonight, I want to share with you the Krog principle. For many problems, there's an animal on which it can be most conveniently studied. And what Krog really talked about was the power of comparative physiology, whereby studying the differences and the unique features of an animal, we can understand the role of a body part and lead to scientific discoveries. Now, one quick example I want to share with you guys tonight is actually of the hibernating polar bear. Now, just imagine sleeping all day, every day. It's the dream. But eventually, your muscles start to waste away. And unfortunately, this is what we see in coma patients. But if we think back to the polar bear, they wake up from hibernation and they're just as strong as the day they went to sleep. So by studying what is unique in the polar bear to prevent muscle wasting, we can understand more about muscle wasting and apply it to humans. Now, my project has nothing to do with polar bears. As a breathing lab, we wondered what Australian animal could teach us about breathing. So naturally, the kangaroo hopped into my mind. So the hopping feature is actually the key defining feature of the kangaroos. But not only do they hop, the hopping actually helps to facilitate their breathing. Now, last time I brought this syringe along because as the kangaroos are hopping in motion, their gut actually acts as the plunger to help them draw air in and out of the lungs. But clearly in such a large venue, that's kind of useless. So, as promised, I brought a bigger syringe along. So you can think of the barrel as your chest and the plunger as everything in your guts. So as the kangaroo hops off, the inertia of the guts draws air into the lungs. And as they land, it crashes into the diaphragm to push the air out. Now for animals that can reach hopping speeds of 40 to 60 kilometers an hour, that's a lot of hopping force being applied to the lungs and the airways. So the main aim of my project is to understand how the structure has adapted to accommodate for this unique hopping-mediated breathing. So right now, I've really focused in on the structure of the airways. We're actually one of the first labs to scan a kangaroo lung in an MRI machine and to isolate out the airway tubes to scan in a micro-CT machine. And we've actually generated some amazing data. We can map the branching pattern of the airway tree, measure the thickness of the airway walls, and even the three-dimensional architecture of the airway wall components. Now, what we currently know in humans is that respiratory diseases such as asthma have associated structural changes. So by studying the structure that's been adapted in the kangaroos, I really believe that is this leap of faith into comparative physiology that can really drive scientific discoveries. Thank you.
Brutal puns, brutal.
Using an Aussie icon, I love it. I can remember when I was pregnant, it got harder to breathe as this little thing was growing inside you, and so there'll be female kangaroos that'll be even harder for them, I'd have thought. Have you studied ones with and without joeys to see whether that extra stuff in the abdomen is having an increased effect?
I would definitely love to move on to that next stage. So we're currently working with the structure first. We're kind of working out the nitty-gritty details in terms of the in vivo studies. Yeah.
Did you really say you had that creature near an MRI?
So not the whole kangaroo, just the kangaroo lung.
Bits of.
Yes, yes.
How did it work?
So we actually went out to the farms and as we know, kangaroos do get culled in Australia, and we actually utilised the tissue which would otherwise be thrown out. So it's a really good opportunity to sample this tissue, and we take it back to the lab, we fix it, and then we can actually put it into the MRI where we scan it and we can study the branching pattern.
Thank you very much. Lovely.
Thank you.
James. So next we have Ben McAllister, also from University of West Australia and the ARC Centre of Excellence for Quantum Engineered Systems. He'll be talking about a subject very close to my heart. There was actually no pun coming on that. I don't know why I paused. There was no need for it. Apart from the organ experiment, shining a light on dark matter. Please make me feel very welcome.
I'd like to tell you a story. It's a mystery of sorts, and it is about space, but it's not science fiction. This is a story about you, and the planet and the galaxy and everything in the universe. These pom-poms are going to be relevant later, I promise. So everybody take one and please pass them around. You see, all that stuff I was just talking about is made up of tiny little particles of what's called matter. Here's some for you guys. And about 14 billion years ago, all the stuff that would later become you and me in the galaxy was all smushed together in a tiny little ball. Then the Big Bang happened and everything exploded across the universe and started combining, making all kinds of stuff which much later became planets and people. Now, that's a pretty cool story, right? But as it turns out, it's really just a very small fraction of the whole picture. You see, all that stuff I was talking about, people, planets, galaxies, that's what's called regular matter. And it makes up just 1/6 of all the matter in the universe. The rest is what we call dark matter. We call it dark because it doesn't interact with light, and we call it matter because it has mass. This is where the pom-poms come in. See, this little bit of white stuff is the regular matter that makes you and me and the planet, and all that blue stuff is the dark matter that we know almost nothing about. So, what is it? Well, when we look at the way stuff moves around in space, we find we can't explain it if we only consider the matter we can see. Spinning galaxies are a great example of this. Most galaxies spin way too fast, and so far the best way we can explain this is that there's a lot more stuff in the galaxies than just what we can see, which allows them to spin a lot faster without stuff being flung off into space. It's like if I spin this ball around on a string. If I go too fast, you expect the ball to break and fly off unless the string can provide enough tension. The extra dark matter literally holds the galaxies together with gravity, and we have no idea what it is. This is the part of the story we don't know yet. So we're literally surrounded by this stuff. Right now, it's passing right through us like some kind of cosmic ghost, and we can't see or feel it. How do you detect something like that? Well, lots of people believe dark matter is made of these little things called axions, which under very specific conditions can convert into photons, which are particles of light, and which we happen to be pretty good at detecting. At UWA, I'm trying to detect axions with what's called the organ experiment. The idea is that dark matter comes in and converts into these tiny little flashes of light that we can then detect. Now, it's extremely hard to do these experiments for a couple of reasons. Firstly, the signals that we're looking for are just unbelievably small. And secondly, there's so much we don't know about dark matter. We need lots of different experiments to look for different kinds of dark matter. And organ is designed to detect a really promising type of dark matter that has never been searched for before. So, hopefully at this point you're convinced that dark matter is important, but if you're not, take a look at the pom-pom and think of everything humans have achieved with just that tiny bit of the matter that we already know about. Computers, medicine, spaceships, and imagine what we could do if we unlocked the rest of the story. Thank you.
I'm going to steal this analogy for my next dark matter talk. Thank you so much.
Pom-pom.
Take it away.
I love the message at the end of that. I thought it was fantastic.
Thank you.
dark matter is very cool. So again, I'd like to know from you, what did the teenagers have to ask you about?
I got some great questions from teenagers. I got a couple of kids who were really worried that dark matter was gonna like blow up the Earth or something. There was this one kid who was like, we're surrounded by it, it's passing through us, is it gonna turn us into dust? And I explained, no, it's very weakly interacting, but if it is the type of dark matter that I'm interested in trying to find, then we might be able to use it for something really positive. If we can convert it into photons, there's lots of potential applications.
I know this is close to your heart, the MC over there, because I think you're working on dark matter too. And
Yep.
there are experiments where you have to go down very deep mines to get there. And I suppose it's pretty dark down there, but is that why you're using these sort of extraordinary sites to try and study dark matter? Can you tell us a little bit more about how you do it?
Okay, so the type of dark matter that you're referring to with these underground experiments is actually the type of dark matter that Alan is an expert in. There's sort of these two...
Guilty.
Yeah, these two camps. There's the weakly interacting massive particles, which are these things that we need to shield really well, so we go deep underground and we look for weak interactions where these dark matter particles crash into the nuclei of stuff that we keep down there. Correct me if I say anything wrong. And then we look for little signals that come out of it. What I'm looking for is this slightly different type of dark matter. They can be sort of complementary descriptions. And with these ones, we don't actually need to go underground. We just need this thing called a dilution refrigerator and a big, strong magnet. So we're actually doing it here on campus at UWA.
So it's dark and darker, is it?
Yeah, you could say that.
Nice. Awesome. Thanks, Ben. Okay, we came up to our final contestant for this evening. Anisa Rohani, Farid, is going to tell us about a culture of open science and data sharing in health medical research. She's taking on the biggest and the baddest of them all, the journals. Would you please make her feel very welcome?
Good evening, everybody. My name is Anisa Rohani Farid, and I grew up in the jungles of Vanuatu, where malaria and dengue fever were killers. Now, this inspired me to move on to do anti-malarial drug research in Australia. So while I was conducting anti-malarial drug research, while I was collecting data in the lab every day, I was confronted with certain forces. On the one hand, my researchers and my peers and professors were telling me we're passionate about contributing to the betterment of humankind. We're passionate about discovering a new anti-malarial drug. Let's do this. Let's discover the drug together. And on the other hand, I was told, Anisa, you need to hide your scientific data because we're in competition with other researchers in Germany. Every day I had to check PubMed to see, have they discovered the drug before me? So the moments in the lab were moments of deep reflection for me, where I asked myself some deep philosophical questions. I asked myself, well, what is science anyway, and who does it belong to? I came to realize that science is a body of knowledge that allows humanity to understand the laws of the universe and to surpass them. And I learned that open science is the practice of making everything in the discovery process fully and openly available, creating transparency and reproducibility, increasing efficiency by allowing new knowledge to be generated in the enlightenment of earlier discoveries. For instance, Louis Pasteur, he discovered that microorganisms could be killed by heat and disinfectant. Now, the sharing of this knowledge has saved millions of lives. And we're also happy to have a Nobel laureate winner in the audience who discovered Helicobacter pylori, and the sharing of this knowledge has also saved millions of lives. So open data is an element of the open scientific process, and data sharing is the practice of open data. Unfortunately, scientists aren't really good at sharing their data because we're set up to compete with other scientists. Now, there's only a particular time when scientists share their data, and it's during crises, epidemics, SARS, Ebola, Zika. During these moments when all our lives are under threat, those are the only moments when we share our scientific data. Now, did you know that about 85% of the world's spending on health and medical research is wasted every year? That's around $170 billion a year. If we shared our data, we could significantly reduce that figure. Data is the foundation of all health and medical research. In fact, data is the foundation of all research. If we share our data, we're unleashing the key to scientific truth. So join me in removing fake news, alternative facts in this post-truth era, so that we can make science more robust, verifiable, trustworthy, and transparent. Science belongs to humanity, to all of us.
I feel inspired.
And you know, there's a wonderful campaign led by Andrew Forrest at the moment to share all the data available for cancer research. Now, other fields have actually been rather good at this, and one of them actually is radio astronomy, again reflecting our MC here.
They are.
Can you tell me, why do you think some disciplines are much better at data sharing than others? What can we learn from our radio astronomy fellows?
Thank you for actually enlightening me with that knowledge. That's very true. Astronomers and statisticians are typically good at sharing data because it's been a practice that they've been used to. They put their data up there, and actually a lot of the data is quite easy to share, you know, and there probably isn't much risk in sharing that data as well, like no commercial value in that data because they're genuinely interested in discovering those laws of the universe. However, when it comes to health and medicine, it's become a very commercialized enterprise, as we all know. I'm not so sure how commercial astrophysics has become.
We'd take $1 if anyone would give it to us, yeah.
But health and medicine has unfortunately become so commercialized. You know, we're told to patent, we're told, you know, when we enter university, we're confronted with certain forces. We're told, you know, open access, make your data open, share your data. But on the other hand, we're told, no, patent, hide your data, be commercial. So my research is specifically looking at changing the reward system at the university level and also working with funders and journals so that we can incentivize high quality research, productive, reproducible, translatable, and shareable research. Because right now the world is in massive crisis, and if only people could trust science, but they're not. Nice.
Yes, well done. Alice Williamson at Sydney, of course, is doing this kind of work on open science in malaria as well. I'm wondering, is this openness moving it forward faster than otherwise it would go?
Yes. So the Open Source Malaria Project, and there are other malaria initiatives that carry out research in the open. So they upload their data live. You know, they're able to interact with other researchers all over the world, which is what I would have liked 10 years ago when I was a bench scientist doing malaria research. So it has significantly advanced the malaria initiative, and and even with Ebola, for instance, when the researchers from Ebola were able to share their raw data that came from the nurses in the Kinima Hospital in Sierra Leone, they shared it to the Massachusetts Institute in Cambridge, and they were able to sequence 99 more Ebola genomes within only a span of a couple of months. So we can see how quickly it can advance when we do share the data, and I wonder, why don't we share all of our data so that we can rapidly advance science and medicine?
All right. Whether Anissa wins today or not, I think she has an incredible career as a future winner of elections with those stirring words. Thank you so much.
Thank you.
And thank all of our contestants this evening. They've done so well. Can I get all the contestants on stage? Can I get the judges to go off stage and deliberate? Because how do you choose the winner? Good luck to you all. Now,
We're choosing them all.
just everyone wins. Everyone wins. All right. Our judges have made their decision. Could I please welcome the judges back onto the stage? Our contestants, hang on, stay. Go on, judges. And I would like to invite the FameLab program manager, Dr. Asta Singh, to, well, say a few words and thank our judges for this very challenging effort. Give her applause, come on.
Now, before I thank the wonderful judges, I'd like to thank the most important people here tonight, which are you, all of you. A big round of applause for yourself to be here, to spend this time with us. Your Thursday evening, you could be watching the cooking show that I don't want to name here. I know. So thank you, everyone. Now, 91 applications, down to 48 through the videos that the contestants sent us into the British Council system. Out of the 48, we went down to 11, and out of the 11, we've gone down to 3. And this is the hardest job, I think, all of us, between all of us, these 3 wonderful people have done. So thank you, guys. Thank you for your time, your deliberation, and thinking over all the aspects, charisma, clarity, and content of the participants. Now, the next and the last and the most important thank you goes to Professor Alan Duffy, right here. Big round of applause for Alan. So thankful, so humbly thankful. 15-year-old daughter has been neglected into the custody of the mother, a very generous mother and wife who has let you be here tonight. So thank you, and thank you all. I'm so pleased that you're here, and enjoy the rest of the night. Thank you.
Thank you, Asa. Okay, so the winners. Judges, I believe you have come to a decision. I can well imagine, but I have your decisions, or at least some maximal number of you that I don't know. Let's just hope it was a fair and statistically robust sample. The audience's, that is, say, the People's Choice Award is Muthu. Round of applause. Oh, you broke their hearts. You broke their hearts. Look at that. This guy. Oh, the glass! No ceremony would be complete without. Let's get that photo. Yeah. All right. Oh, and there's more. There's more. Just hold both. No, get your photo with both of the prizes. Come on. And keep the round of applause going. Very well done. Thanks so much. And now to announce the runner-up. Helen, I believe you have the envelope.
Okay, everybody. The runner-up for the National 2018 Finals in Perth of FameLab is Toby Hendy.
For the winner.
Is it La La Land?
Oh, please, please.
Oh la la. And the winner is?
Not to be sneezed at, Vanessa Girotta.
Woo! All of you, move forward, move forward. And a giant novelty check. You get a novelty check. And if I get everyone to stand forward, clear the danger. And of course, no win would be complete without a giant novelty check. Another round of applause. Oh, wow. That's it. And that's it. That's the national finals. That's it. It's going to compete in the internationals at Shenton Festival. She represents Australia. Thank you all for coming this evening. Thank you to all our sponsors and partners and our judges, Asa, the team, everyone else. Thank you so much for coming.